Hello, this is Zishan from Icahn School of Medicine at Mount Sinai. Now I would introduce my project, Master Regulator of Protein Abundance across 6 cancer types. As we all know, the central dogma defines the transmission of genetic information from DNA to RNA, called transcription, then from RNA to protein, called translation. Previously, more attention focused on the transcription process, such as TCGA project provide mRNA expression profile for a large analysis of transcription. Now we focus on the translation process, where there is a lot of post-mRNA process modulating translation rate, protein transport, or protein degradation, and so on. However, how this process manifests in cancer remains largely unknown. Recently, high-throughput output mass spectrometric technologies have yielded quantification of over 10,000 proteins across many cancer types. CPTech project. This gave us an opportunity to explore translation regulation. Thus, we would predict translation regulator and hypothesize translation regulator mediate translation of mRNA to protein. Using 6 cancer cohorts with sample paired transcriptomic and proteomic profiling, we predict translation regulators and validated with known gene site regulating translation. Here is the workflow of our project. Briefly, we predict translation regulation based on a 4-step computational pipeline and wrap the pipeline into an R package. We use known function gene site to validate our predict translation regulation and revealed functions regulated by translation regulator. We would introduce them one by one. First, let's see the data. We obtained samples paired mRNA protein expression profile of 791 samples across 6 cancer types from CPTech project. Basically, each cancer type involves thousands of proteins and hundreds of samples. Next, I would introduce the 4-step computational pipeline to predict translation regulator in each cancer type. First, we construct co-expression network at both protein expression level and mRNA expression level based on the Spearman correlation. BH corrected p-value of less than 0.01 is defined as significant, which is also applied to other steps. Second, we remove the mRNA co-express network from protein co-express network, generating protein unique co-express network. Third, we used semi-partial Spearman correlation to generate a direct graph of translation regulation. Fourth, we permuted 10,000 times for the directed graph of translation regulation and predict translation regulator based on the number of targeted genes regulated by candidate translation regulators. The round of permutation is repeated 30 times, and the overlap of translation regulators of these 30 times were our predict translation regulator. Here is our predict translation regulator across cancer types. There is thousands of translation regulators for each cancer type, except for ovarian cancer, and some translation regulators shared across different cancer types. Next, we have validated our predict translation regulator with known functional gene site across different cancer types. As shown on the picture, iron binding protein is significantly enriched. Interestingly, nuclear pol complex is also significantly enriched, indicating it may play important roles in translation regulation process, and we also observe there is no overlap with transcription factor. Then we also have got similar observation for translation regulators shared across cancer type. Then we have inferred translation regulations based on the binding region of iron binding protein from the ECLIPSE dataset of ENCODE project and validated ours. And we found our predict target of some translation regulators are significantly overlap with translation regulation based on the ECLIPSE dataset. Then we have picked out some translation regulators for further experimental validation, which is still undergoing so far. Next, we performed functional enrichment analysis and found that splicema pathway is the top pathway regulated by translation regulator in cancer. Lastly, we wrapped our computational pipeline of predicting translation regulator into an R package. User can easily predict translation regulator based on their own sample paired protein mRNA expression profile by using two functions, network and MATR. That's all. Thanks for your listening and have a nice day.